In this video, we are going to create a model of homologous chromosomes and then simulate them going through crossing over during prophase 1 and meiosis 1. Here's a list of the supplies you will need. If you haven't seen my mitosis and meiosis modeling video, you might want to check that as well. I go through all the phases, and I also do include crossing over in that video. However, in this video, it'll be on crossing over only. So let's get started. Please take 2 pieces of construction paper, each of a different color, and place them on top of each other. Then I want you to fold and crease them over top of each other in half, and then do that one more time so you fold it in half again. Once you open it up, I want you to cut along all creases so you get 4 equal-sized parts. Well, when I say 4, I really mean 8 because you had cut out 2 sheets of paper on top of each other. Then take just one set of that square, like one on top, one on the bottom, and fold it long ways down the middle so you have a nice crease. Next, draw a big, kind of like a bubble letter X, if that makes sense, but just half of it. So another way to look at it would be like skinny, skinny butterfly wings, just half. And the key thing when you're drawing out this shape is you want to make sure that along the crease is where you're leaving a spot that when you eventually cut this out, there'll be a connection point. You want them to turn into 2 identical size X's is basically what we're going for here. Then actually cut it out, and you should end up with 2 identical shaped X's. But of course, they're not really X's, they're representing chromosomes. And what we're going to be doing now is we're next going to label genes on our chromosomes. So on one of your chromosomes, just on one side of it, I want you to write a series of letters, whatever letters you want, and I want you to separate out each letter by a horizontal line, because this is representing individual genes at their own locus. When you're finished drawing out all the genes at their own locus, meaning locus means location, a location, then on the other side of the same exact chromosome, I want you to rewrite all of those same letters in the same form. One thing you have to be careful of is please don't use all uppercase or all lowercase letters. Try to mix up uppercase and lowercase letters. Why? Because an uppercase letter represents a dominant allele or form of a gene, and a lowercase letter represents a recessive allele or form of a gene. And it's just not likely that from one of your parents, or really either of your parents, that you would get a chromosome with all dominant alleles or all recessive alleles. So there's going to be a combination, a variety of different dominant versus recessive. So what about that other chromosome of the other color? If you notice, both of these chromosomes are the same size and shape, and that's because we literally folded paper and cut them out to make sure they were. But in reality, there was a reason we did this. You actually have 46 chromosomes. Out of your 46 chromosomes, 2 of them are your sex chromosomes, either XX if you're a female or XY if you're a male. But all the other 44 come in what we call homologous pairs. And a homologous pair means you got that chromosome from your mother, but you also got that same chromosome from your father, just with different information. When I say different information, I don't mean completely different information. It has the same genes at the same loci or locations. It's just which variation of the gene can be different, and like it's going to be because they're not the same person. So on the other chromosome, we want to look at the letters that we wrote out on our first chromosome and use those same exact letters, but mix up some of the uppercase versus lowercase to demonstrate that when you get, let's say, chromosome number 1 from mom, and then the homologous chromosome number 1 from dad, that it's really a copy of the same chromosome, just different forms of the information. So for instance, my top gene, I wrote a capital E on my pink chromosome representing the chromosome from the egg, and then on the blue chromosome, I wrote a lowercase e representing that letter coming from the sperm. So the idea here is, let's say capital E is referring to the ability to roll your tongue. That's a dominant allele, a form of a gene that's dominant. But let's say the lowercase e is representing the inability to roll the tongue. So it's the same idea of the gene location. The locus is representing whether or not you can roll your tongue, but it's a different version of that gene from your mom versus your dad, or whatever the relationship is, we're talking about the egg that you came from compared to the sperm that you came from. So whatever letter by letter you wrote on the pink chromosome should match letter by letter what you wrote on the blue chromosome. And I'm saying blue and pink because those are the colors I use. But in terms of your two different pieces of construction paper chromosomes, but you have to be careful that they're literally identical on the same color. So I had capital E, lowercase m, lowercase I, capital L, capital Y, lowercase s on my pink one, but then I had lowercase e, lowercase m, capital I, capital L, lowercase y, capital S on my blue one. But again, both sides were identical. So the deal is, the reason we have X-shaped chromosomes here, these homologous pairs are in the shape of X's, is because prior to going through meiosis, they went through interphase in the cell cycle, and within interphase is the S phase, or synthesis phase, where DNA is replicated or synthesized, and a duplicated set of the gene information, the chromosomes, the DNA, is made. So a regular chromosome is really just one of these pieces, but right now that their X is connected in the middle, we refer to either side as a sister chromatid, and we refer to the whole X-shaped structure as one chromosome, which is complicated, but that's because they really count chromosomes based on their centromeres, like the center location, which right now they're connected. Now, when these get disconnected during cell division to go to opposite cells, they have their own middle point centromere, and they would be considered their own chromosome when they're physically separated from one another. So it's really interesting and kind of complicated about prophase 1 of meiosis 1 is that homologous pairs line up next to each other. And again, when we say homologous, homo as a prefix means same. So just think these are chromosomes of the same size, the same shape, the same basic genetic content, though different variations of the genes from the sperm versus the egg that this person came from. But they literally line up next to each other and start interacting and swapping genetic information in a process called crossing over. So what we're going to do now is I want you to take your chromosomes and start visualizing how they might actually kind of cross over with each other, where different sections of the chromosome overlay on top of each other in a way that they can swap genes at a specific gene locus or location. And then I want you to take your scissors and literally cut off a particular gene. Just make sure it's the same letter. It doesn't matter if it's upper versus lowercase. But for instance, I cut off the letter S on both of the homologs, which is the way you refer to homologous chromosomes as well. And I'm going to swap them around and tape those genes in place because recombinant DNA has formed, where the maternal and the paternal chromosomes literally swapped a gene at that particular locus. That's what we're visualizing here. And because it's random how often and where it'll crossing over will occur, I usually like to simulate at least one other gene crossing over because I think it's kind of fun and it helps us understand that there's no rhyme and rhythm. It can be anywhere, anyway. The biggest thing, though, is it has to be swapping genes of the same locus or location. So if you notice, I swapped out the letter M at the top to represent my other location. But again, it can happen multiple locations. It doesn't have to happen a certain number of times. But why would this happen? Well, it increases genetic variability in the offspring. Because keep in mind here, we're in meiosis, and the end goal of meiosis is to create gametes or sex cells. So in conclusion, let's just talk about a couple of other terms to know. So when the homologs line up next to each other during prophase 1, it's a process called synapsis, the actual lining up. Each pair of homologous chromosomes is a tetrad, and then basically while they are next to each other, they can swap this information like we were talking about, that term called crossing over. And when they recombine with the new genes at the new location, the swap genes, it's called recombinant DNA. Well, I hope this was a helpful video. Construction paper biology. Like, share, subscribe, oh, won't you please? Thanks!